You guys able to open it?
I
Anybody
can't
found
find it.
No,
Did you say it's under quizzes?
it's under quizzes. You guys see it now? Yeah, anybody found it?
Yeah.
All right, cool. By yourself, guys.
She's trying to find the bright
Oh.
space.
Yeah, my
on
computer
Safari.
died, so.
All right, everybody good to go? Any more time? No? All right, let's get started. The thing has a timer, so I think like within the next minute or so, it's probably gonna kick you out, so submit your answers. All right, let's do this. All right, so Wednesday? We were talking about microtubules, right? For that one, big thing is we have this fiber, it's dynamic, that's a big important thing. The other one is, remember, we have two different motor proteins, right? One of them likes walking towards the positive side, the other one walks towards the minus side. Using that, it's a really powerful tool for us to move things around. We can send them in specific directions. Today, we're gonna a little bit go into the endomembrane system. We're gonna take a little bit of advantage of that ability of this microtubules and those motor proteins to move these vesicles. But now the big thing we're gonna be talking about is gonna be about when you're forming that vesicle, and you're saying the goal is to send some molecule or some protein transcription factor to this other site, how do you ensure that you're sending that molecule in that direction? And what you're gonna see is it's, again, that protein-protein interaction, which helps trap the molecules. We bring them into these vesicles, and then we can send the vesicle, and we can drop them at whatever target we want to send them to. Yeah. So that's gonna be the big theme, so just keep an eye out for these guys. All right, so again, goal, we're gonna be understanding how the proteins are moving through these subcellular compartments. And then we're also gonna see how they get made inside the ER itself. Wait, that's not right. There we go, that looks better. You're gonna be looking at how the protein gets made. There we're gonna look at two really cool types. One is like, if you make a protein, you can dump it into the ER. In the other cases, you can make the protein, and based on how you use that little molecule, you can embed it into the membrane itself. So it's gonna look pretty cool. So let's get into this guy. All right, so first, let's orient ourselves, right? Here in our cell, we're looking at eukaryotes, right? What's the big features that we're looking at when we're talking about eukaryotes? What's the biggest one? Biggest feature, working out. Yeah, we got our nucleus, right? Here we hold our DNA, the DNA is transcribed to RNA. The RNA then basically leaves the nucleus, and then it can go into two different places, right? It's gonna hit a ribosome. It's either gonna be a ribosome that's gonna be on the ER, that'll be a rough ER, or it can hit a ribosome that's floating around, and that'll give us basically those like cytoplasmic proteins. Once we've made that protein we're gonna be talking about today, we're gonna be looking at that really dark purple one, right? How we're making proteins in that domain. After we make it, we then, as we're doing modifications, we're also prepping it to send it to its compartments. There, we're gonna hit this guy, right? The Golgi apparatus. There, if you guys remember from before, it has a cis side, which is closer to the nucleus, trans side, which is further out. These vesicles basically kind of link it up to the next site, get you bring the protein to compartments, to further modify that protein, sugars and stuff. And at this other side, you basically reduce the vesicles, you can send it to a lysosome, you can send it to a plasma membrane, or you can send it to any other organelle. So let's look into that, right? That's the picture. This is basically the map of what I was talking about right now, right? So, again, we're starting in the nucleus. This is where we're sending our, as the DNA sends the RNA, and that's what lets us get us to the ER, right? ER, we're making that protein, sends it to the Golgi. There's going to be vesicles that move backwards and forwards in that place. There, you can think about how we're using the microtubules, the dynein, and the kinesin to basically move those vesicles backwards and forwards along the same microtubules, right? After that, within that Golgi, you can see that we can also send proteins depending on how we send the vesicles, right? We can send it to the endosomes, which eventually get produced as lysosomes. We're going to go into details about that today. The other thing is, like I said, these vesicles, we can send them all the way to the plasma membrane and basically kick them off, kick them out of the cell. And then this will be like those, you can go through a secretory vesicle. At the end of the day, you can dump it out outside of the cell. So big thing, vesicular transport. Yeah.
Why is late endosome above early endosome? Like, why is it named like that?
So that one is because the endosome is like you're trying to grab something and eat it. Actually, you're bringing it in. So it's like this is early stages, and as it gets further in, it's like it's maturing.
So the early is like the mouth that eats it?
Yeah, if it's a macrophage and you just ate something, it's gonna bring in that vesicle and it's gonna bring it in. Once you bring it inside, then you kind of basically mess with it. I can't remember if it's pH related and then there's extra enzymes. And as you bring those in, it's gonna basically start eating the things inside of it. So you can break down sugars, you can break down the lipids, you can break down proteins. Okay. I'll look up what the enzymes are, because those are pretty cool. Like they get like regulated in the compartment, so this catalytic or this catalytic function, you don't want it everywhere. So you can only try to pocket it into a single thing. So I'll bring those up. But today I only prepped it for the lysosome. All right, let's go watch a little video about the story. You guys said you guys are able to click into the videos, right? Yeah, all right.
All protein synthesis in eukaryotes begins in the cytosol, where ribosomal subunits come together and begin translating an mRNA near its 5' end. Soon after the protein starts to emerge from the ribosome, it may become directed to the endoplasmic reticulum, or ER. It is targeted to the ER if it is ultimately destined for incorporation into the ER, the Golgi apparatus, lysosomes, or plasma membrane, or if it is destined for secretion. In mammalian cells, most proteins enter the ER co-translationally. These proteins have an amino acid sequence called a signal sequence, which is typically located at the amino terminus of the polypeptide chain. Signal sequences span about 20 amino acids and include a stretch of hydrophobic amino acids preceded by a basic amino acid. As the signal sequence emerges from the ribosome, it is recognized and bound by a signal recognition particle, or SRP, consisting of 6 polypeptides and a small cytoplasmic RNA. The SRP binds the ribosome as well as the signal sequence, and in so doing, stalls translation. The SRP targets the entire complex to the rough endoplasmic reticulum by binding to an SRP receptor on the membrane of the ER. GTP molecules bind to the SRP and the SRP receptor. The GTP binding triggers the transfer of the signal sequence from the SRP to a membrane channel called a translocon. Hydrolysis of GTP to GDP leads to the dissociation of SRP from both the receptor and the ribosome mRNA complex. Transfer of the ribosome mRNA complex to the translocon allows the signal sequence to interact with short hydrophobic side chains located in the narrow neck of the translocon channel. This interaction opens the translocon by moving the plug away from the translocon channel. As translocation proceeds, the signal sequence is cleaved by signal peptidase, and the polypeptide is released into the lumen of the ER. Thus, the process of protein synthesis directly drives the transfer of growing polypeptide chains through the translocon and into the ER.
All right, so right now you guys saw that we're going to be catching we're basically looking at this side. This is a drawing for a little simplified version, right? But the mRNA is coming out, we're catching it with the ribosome, right? That SRP, you guys should be very comfortable with that, how that one works. I'm gonna get into a little bit later, but when that one comes in, stops translation. So it has, like an SRP has multiple functions, right? One of them is gonna stop this thing from happening. The other one is gonna help us find the receptor. So it's gonna be like an anchor to bring it to the edge of the ER, right? Once we're doing that, you guys can see that the sequence itself has important features, right? One of them was that hydrophobic region, right? That's what led to basically travel through this part of the center of this of the translocon. The translocon has a bunch of alpha helixes, which it uses to kind of be able to sit through the membrane by having alpha helixes that are hydrophobic. It can sit across this dark orange hydrophobic region, right? So when we push this, this allows the hydrophobic region to come in. We can start getting it into the inside. If we want the protein to be released, we have to actually cleave that first alpha helix that came out of it, and then it can get into the free protein. If we don't cleave it, that's how I'm gonna show you we're gonna start making those transmembranes. All right, so now we're super zooming in. Again, that SRP molecule, super interesting and has to do a lot of different tasks, right? So here we're looking at the first domain. So you can see in blue, we have a mixture of our RNA molecule, and we have a mixture of proteins, right? This thing's working all together. And in this part, this is what we can use to actually stop translation, right? So you can see ribosomes floating around, mRNA shows up, it's trying to start translation. That's the thing that's basically gonna jam into it and kind of stop it, right? Then you have the hinge, but then this is the other one, the signal sequence binding pocket, right? That's how it's gonna recognize what mRNA during translation should it stop. If it has the sequence that we need for something that has to go into the ER, that sequence is just gonna basically show up right here and it's gonna get grabbed onto, right? So if you put this in front of a protein, this sequence, the sequence is your basic. If we add it to any protein that we want to study, it'll do this mechanism. It'll catch it right here. Once it gets caught, we can grab it and this thing will carry it to the ER and it'll drop it into it. Right. So that's the big power of the sequence, but again, it's coming from this guy. The other big task you guys gotta feel comfortable with the SRP is it helps interact with the receptor, right? And that's the big mechanism of let's get it in there. So then here's basically our storyline, right? Here's your ribosome, here's your mRNA, here's the N-terminus is trying to build your polypeptide. That's your protein machine. There are SRPs going, recognizing that tag, and saying, hey, this thing has that hydrophobic pocket. It recognizes that sequence, grabs it, and it's going to stall translation until we get it to the membrane. Right. Here again, that SRP is giving us that ability to recognize the binding to its receptor. When we're looking at that interaction, I want you guys to keep thinking like we did when we've been talking about ligands and receptor, that specialized shapes, they grab onto each other like a specialized handshake. And they have high affinity and they grab to each other very well. At that point, you guys saw it goes from GDP bound to GDP bound. That's basically going to change this conformation, right? That release is going to help us get that ribosome to start working again, so translation starts. This thing basically is kind of anchored at that spot. And as it's making the protein, it's just basically shutting it across the translocon. That's what they were saying, right? It was giving us pressure or force to make the plug open up. At that point, you can just pump the protein into the ER itself. After that, we're going to cleave it, and then you can release the protein, right? So these are really good for if we're gonna make, let's say, a soluble protein that's just basically floating around in the ER. If you don't cleave it, again, that's the big thing, you don't cleave it, technically this thing's gonna be stuck for a bit, stuck with the translocon, but I'll show you a little bit later how we can release it and it'll stay stuck inside the plasma membrane. And then we can trap protein across the membrane. So here we go. Same mechanism in the beginning, right? Here you can imagine we would have had to draw on upstream the ribosome, the mRNA, the protein being made, the SRP grabbing onto it, bringing it to the receptor, you set it up just like before, you end up at this. Right? In this case, the goal is to make like a tyrosine receptor kinase. Remember, those are 3 domains, and they only have 1 alpha helix going through the plasma membrane. There you basically have a region that's hydrophobic, right? That's your hydrophobic alpha helix. In the translocon, you guys kind of see here in this 3D shape, it's like a channel, and it's a bubble, but it has the ability to open up. When you open it up, what's going to happen is this protein kind of doesn't want to go into this environment, because remember, for the most part, it's like, this is water, so it's going to be a hydrophilic environment. Your hydrophobic stuff doesn't really want to interact with it, so you won't be able to push it out. That's when basically it pops off to the side. It opens this guy up, and then you can release it into the plasma membrane. Right. For these, you're going to find that you're going to have that hydrophobic alpha helix, and you'll probably find positive charge molecules right outside of that. That way, they'll feel comfortable with the phospholipid, right? The charged heads. So, big player here. And then the other cool part is, here in this case, it's a single loop. So at that point, this ribosome becomes loose, and you can see it just basically floats off, and it can make that cytoplasmic part of that protein. Some of these are pretty cool in the sense that if you want to make it a multi-pass protein, what's going to happen is, as it's making it downstream in here, you'll again have another one of those sequences that the SRP will recognize. And what happens is the SRP will show up, it'll just try to stop it, it'll bring it to a grinding translocon, and it'll dump it in the next one. So that's pretty cool. Let's see, I have a picture for that one. Sorry, get out of the way. Out of the charge. Yeah. So, sorry, this is the one I was telling you guys about. Like, it likes to leave a positive charge just to be happy with the phospholipid heads. But here's the way we can make our multi-transmembrane segments, right? So if you want to go in and out of the plasma membrane, it's, you get the first one in, you get it to pop off. In the one I saw, they were basically, not that I was looking at this one, but for this one, it gets popped off, it'll be loose again, another SRP catches it, and it re-signals to stop translation, brings it to the plasma, brings it back to the edge of the ER. It dumps it in, it does the same thing, you're gonna have make a loop, and then it produces protein. Based on like how you look at the protein, they'll be set up different. You see here the different color codes, like the yellow and the dark yellow. If we looked at the sequence of the mRNA 5' to 3', if we were thinking about on what side you're gonna grab the SRP, it's if you grab it on the 5' side, right? And you do this part of the protein is basically gonna get shoved into the ER, right? On the next one, you basically have to arrange these in different orientations to get the different passes, right? So this would be, you get it into the protein, this will be on the ER side, you make another loop, this will be the cytoplasmic side, you go to the next one, right? You get back, you can do the ER side up until you get to the next sequence. So by having these spaced out, you'll have them arranged so that you can actually get your alpha helix, a loop inside the ER, an alpha helix, the other loop on the cytoplasm. You can even put another alpha helix and get the cytoplasmic side. Like this one, you can see it's really, it's like really dramatic when you look at the G protein couple receptors, because they have to have 7 passes to get them to work. And the crazy part is, as you're making these loops out here and out here, remember that GPCR is using those loops to make one of those receptors, which means it's like a super 3D shape to be able to catch like epinephrine, right? One of those small signaling molecules. On the inside, it's using all these loops to be able to have like a GEF. And then we can play with the G protein. So it's super fancy, super generic. I think these guys are really fun because of that. All right, so now we've made the protein, and we basically made two versions now, right? Imagine this is my rough ER. We can make proteins, and release them on the inside, or we can make proteins and kind of make them sit across the plasma membrane like that. So now how are we going to enrich this or send it to the right environment? Let's go back to that endomembrane stuff, right? So in this case, imagine there we're inside the ER. The red's going to be the protein that we're kind of keeping track of. Would be these guys. The green term you can use to represent basically this kind of thing of one of these. When they're doing this, can you remember last time I kind of drew it for you guys? You can have this thing butt off, right? You start folding the plasma membrane around the ER. You basically make a little bubble, and then it'll pinch off on itself. And whatever the contents inside, you normally get closer with receptors, but I'm gonna get into that. But once you've made these bubbles, you basically here are using the microtubules, and you use your motor proteins to move you away from this and get you to the next compartment, which for the most part is going to be your Golgi, right? You have your protein, you add a little bit of sugar. Once you get to the Golgi, you're going to add a lot more sugar, but you're going to send it to these compartments. If we zoom into how they do that, what we're going to find is that there's basically going to be things that within those vesicles will actually try to catch our protein. So it'll help us enrich it. Because otherwise you'd be wasting a lot of energy making vesicles, and it'd be like whatever diffuse molecules you have, we just grab them and throw them away, like trying to send them forward. But instead, by having like little receptors, like in this picture, you can look at like basically the transmembrane protein that's going to be hitting like our little receptor, which is catching our particles. These, they have a way to get enriched on that region of the vesicle, but they're going to trap these red ones, which is the protein that we care about. Right? They'll grab onto it. This thing has a really cool mechanism of like it deforms the plasma membrane and then it pinches itself off. So at that point, you trap that high concentration of the molecule we care about. Right? Send it through the microtubules. Use your motor proteins, carry it to the next compartment, and boom, we're going to get started getting ourselves to the Golgi, right? At this point, what's going to happen is you can start recycling these guys, which would be like, sorry, here, these will keep kind of combining with each other. But at this point, you see you can cause the release, right? Your protein of interest, the red one that we care about, you're going to release it from the receptor. This protein can get further modified, and it's going to keep going down its pathway. But the receptor itself, we can actually grab those, and we can actually send those back to the ER, right? So it'll be almost like a transporter. Grabs the protein we care about, traps it, carries it to the Golgi lets go, and the transporter comes back. So you can actually reuse the same molecule over and over again. So for this one, you guys should feel pretty comfortable telling me this story, right? Comfortable with how the receptors help us out, how they're being recycled, how we can get a protein that we care about and send it to the following compartment. And that only goes forward. And that's because on the way back, you can see this basically, that's our receptor, the K-del. It basically competes for that side. So instead of being able to grab onto the protein we care about, it grabs onto something else, and that's going to be able to send it back.
So do those little bubbles just go back and like absorb into the ER and become part of the membrane again?
Which colored?
Like you're saying the K-del sends it back. Does it just bubble back into the ER?
Yep. So it'd be like
So it's kind of like recycled.
exactly. So it's a good analogy for this one. Because the plasma membrane itself, it'll pinch off, grab the bubble, and inside you'll have the carrier. Sorry, you have the carrier, and inside you have the protein you care about. That bubble goes, when it hits this compartment, it's going to do this thing where it'll plug in and it'll dump itself like that. When it's doing that dump, the protein we care about is going to go loose because this thing's going to bind to it. Right? Now that this binds to it, this one you can send this back. So there's like basically a transporter, and this is like a temporary kind of holder, so it doesn't interact. Once we send it back here, it does that merge again. Whatever's in the inside of the Golgi ends up on the inside of the ER, and vice versa. Once we get in here, here's when we break the interaction between the K-del and the receptor, and you basically reset it so now it can grab onto the molecule. So I'd like to ask you to tell me about just The KDEL receptor, you should be able to tell me a story of how it's cycling. If I ask you about a protein leaving the ER that we care about, then you should tell me how the KDEL helps it on this side. But the goal is that we've enriched the protein onto that, to the Golgi. But all of this, in the drawing doesn't say it that well, but the thing is, again, I want you to think about those transporters. They're using a really fine protein-protein interaction. So it's those handshakes of like we specifically grab onto a molecule and we carry it over.
How does the KDEL get back to the Golgi?
I think the KDEL gets thrown back into vesicles. But it's not gonna be put into it. I don't remember what's the modification to it. I can't remember exactly what. It's like sensitive to the environment, if I remember correctly. So it's like, in this environment, it favors the interaction. Once you get it back there, you lose the interaction between those molecules. I don't wanna say what it is, because I might say it wrong, but that'll be one of my, put it on your question, I'll look it up. Same thing with you, if you put that as a question, I'll look it up. Yeah. Well, yours is different, but put yours up. All right, so part of that, the thing I keep telling you guys about, like, when you have these different receptors, the receptors have like these secret handshakes with each other, of how they recognize each other, and they're very specific. People have tried to study how these proteins work. The old school way of doing it is you'd go in and you'd break your protein into chunks, you see what protein went to the right environment. And you would get smaller and smaller fragments to say, this is enough of that amino acids to be the code for that special handshake. So in this case, this is basically the summary of the work that all those people have done, right? So for this one, the way we're gonna read this chart is, this is like what they've minimized to say, this is what we believe the signal sequence is. And then you kind of read it based off of these facts, right? Which is, what are the properties of those amino acids that define them? Positive charges, negative charges, are they hydrophobic, or does it like to get hydroxylated? Getting OH groups with them. And as you can see, if you want to send stuff to the ER, right? There's our hydrophobic signal we've been talking about, right? That's the way that our receptor grabs onto that. If you want to send something to the mitochondria, we basically space out these positive charged amino acids, and that's enough to get the signal to go to that domain. And it's going to do it just like the other ones did, right? When we're bringing in the vesicles, those vesicles are destined to their specific compartment. As you do that, you have a receptor, recognizes the sequence, grabs onto it, and it's basically going to drag it to where we need it to go. That making sense? Yeah? All right. Yeah, I brought a lot of this one, import into the ER. So I wouldn't ask you to be like, hey, memorize that it's leucine, valine, but I might give you something where I talk about like, you have a hydrophobic region. We think it's a peptide. Where do you think it would go? The other one I'll just as to mess with you guys, to test you. I'll look at this one, which is, if we want to import something into the nucleus, what are we doing? What do we have to do to that protein, or how do we tag it?
A hydrophobic section.
Sorry, the color's bad. It's the lysines are supposed to be red.
Is that positive? Oh, positive.
Positive, my bad. It should look better on your guys' screens, right? A little closer.
Oh, yes.
Yeah, so it's red. So that means that, yeah, if I tell you, hey, we have a stretch of 5 amino acids and they're positively charged, I wouldn't give you the amino acid code, so you don't have to like memorize all the amino acids, but you would say, hey, if I see a positive charge, it's gonna go into the nucleus, right? And then the last one, so I'll do import to ER, I'll do the nucleus, and then let's see what's a cool one. We'll do an import into a plastid. How about that? Right? So import a plastid, what are we doing?
Hydroxylated.
Yep. So if I give you guys a hint that, hey, we have this hydroxylated sequence, where do you think it's going? You guys should be able to use that table really fast and tell me it's going to this compartment. Yeah. Or the other one is, what would happen if I'm trying to do, if we find this protein, we find that it's hydroxylated in one of the regions, and I cut it off, what do you think would happen? Or, my bad, let's do it for this one. Import to the ER. If we have a protein, we find this right in the N-terminus. Hey, I found that hydrophobic region. I chop it off and I have the rest of the protein. Where do you guys think it's gonna go?
It's gonna be destroyed.
All right, that's one hypothesis. Do we have any takers? So destruction. One more hypothesis.
If there's another signal on it, would it send it to the other place?
Hypothesis number 2, right? There has to be another signal. I'll take one more hypothesis.
Chaperone, like they will modify it to be functional. Like the chaperone, they will modify it to be functional.
But where would it go?
Okay.
So I'll take your first hypothesis in the way good thinking, right? It's like, hey, if you chop up a protein, what was your first hypothesis?
That will be destroyed.
Yeah. Some of the proteins are very sensitive. You try cleaving something too big, it might make it unstable. It wouldn't fold properly. You'll send it to destruction. So yeah, it's a good hypothesis. It's a likely thing to happen. It happened to me. I tried doing an experiment like that. Months of my life, I was like, I wasn't getting any good plants, and it was just like, you can't cut stuff off. So you're right. That's a good hypothesis. Your hypothesis was?
If there's like a green section and a red section, and you chop off the green section, the red section will tell it to go what matches the red signal. So like import to ER has green and orange.
Mhm.
And so if you cut off the orange section, the green section's gonna tell it to go somewhere else.
Perfect. So whatever other signal we have, that's what's gonna drive it. If we have a signal that you actually said right now, something like it's blue region, or we have a red region, or something else, that other signal is gonna override our mechanism and might send it to that compartment. If it doesn't have anything at all, we're just gonna make a cytoplasmic protein, right? Because remember, the SRP slows it down and tries to put it into the ER. At this point, we're gonna be making, I'm sorry. Remember, the SRP recognizes the signal to get it to the translocation to get this whole thing to happen. If you don't have that signal, you're gonna keep making that protein the cytoplasm. At that point, to have a cytoplasmic protein, technically you can only go here. But that would have put the answer I would have put on my key. If you two would have argued your points, I would give you full credit for that, right? Because it makes sense. You're like, hey, protein's gonna be most messed up, it gets degraded. I normally update my key and I'll put your answer for it. Same thing with you. So both of them are good. Thanks, guys. Put that on your, put like +2 on your note cards for that one. Both of you guys. Good puzzle, guys. Let's go back. Where were we? We did this one. Let me check out this video real quick and then we'll be back, guys.
Question.
Yeah.
So to import to the ER, it has to be hydrophobic, positive and negative? Or like, how does that determine it if it goes into the ER?
So,
Like if it weren't for hydrophobic, like it would go somewhere else?
yeah, because remember the SRP? The SRP, like, it has this part of the SRP, it's actually helping to recognize that sequence, right? So if you can't grab onto it, all the other tasks that we have up here, we lost, right? We're losing the translational pause. That means you're gonna keep making that protein in the cytoplasm. What's the other thing it does? The SRP does that whole task of like, right, wrapping up to the hydrophobic, we bring it to the receptor, remember the receptor setting it up with the translocation to push it across this. The protein's not gonna be able to make it into the ER. So it's gonna be a completely different confinement. Yeah. Cool.
What happens to the rest of the protein that's like cleaved off of the protein that's released, like that's kind of stuck in there? Like does it code for anything, or is it just like to guide it?
It's just to guide it. There's gonna be a lot of them that their whole role is like, you want it to get them to a certain compartment or get them to do a certain task.
Mhm.
And that thing will just get them there and then you cleave it off.
Okay.
I think you guys have seen another case where we've seen something like this. It was when we did the protein folding, right? There was the 3 pathways. There was the IRS, there was Ann's favorite, and then there was the last one. Remember that one? It makes the protein, traps it on the plasma membrane, and it gets cleaved, and then becomes the nuclear factor gets released. So
Okay.
you can do it right now and get it to work, or you can do it later.
Okay.
Cool. So my bad, back to your question. So hydrophobic part, we're done with. The positive charge, which is the other part, it helps us with this. Just to keep it happy on the plasma membrane. If you don't have it, your protein might not be too happy because you're gonna have like maybe hydrophobic amino acids or trying to interact with the phosphate groups and they're not gonna get along, right? And I think the negative charge, I think it helps with interaction. But in order of functional wise, if you hit one of these two, you might break that so it needs to work just a little bit. If you take out this basic thing, so the hydrophobic part, you're probably not gonna get it to work at all. So the bigger the mutation, the more drastic the result. I got video and then we keep going.
The endoplasmic reticulum, or ER, after which they proceed to their
All right, you guys watch that one on your own. I think it's gonna be a slow one. All right, so we're back. So now we're gonna be talking about how we're gonna be moving things from different compartments. Again, we're not showing it in the drawings, but in the back of your head, I want you guys to keep in mind microtubules and then the motor proteins.
Can you go back one slide to the table?
Yeah.
So on the, so for example, the import to ER,
Mhm.
there's the negative and the hydrophobic.
Mhm.
But what determines whether it's cut and then like the hydrophobic part will go to the nucleus, or if it's like the full? Does my question like make sense?
Yeah. So the translocon and this and then the peptide we're importing, they kind of get set up. Again, those hands, the handshakes with the peptidase is going to come and cleave it.
Okay.
So that'll direct us and bring that peptidase to come in and cut this thing off at that sequence. Normally, if I remember correctly, it's like when you're pushing stuff to the translocon, it itself has hydrophobic alpha helixes. So when you're trying to like jam protein through it, if it's hydrophilic, the interaction isn't happy, the protein kind of gets like, it goes right through because it doesn't want to interact with it. It'd rather interact with the water inside. So that'll push them really fast. When you find anything that's hydrophobic like this, it'll be more stable to stay stuck inside of that than getting kicked out. So normally that's what ends up kind of like acting like a small break or like it traps it for a little bit.
Okay.
In that trapped time, it's the best time for your peptidase to come in and cleave it off, right? And at that point, you can cut this thing, release it, and the rest of the amino acids are still coming out from the ribosome. Most likely that's when you're doing all your hydrophilic stuff, and you're basically pushing it right through. Like it'll slide right out.
Okay.
Yeah. But you can play with the order. I remember that one was pretty cool. We were playing around with experiments of like, these have spacings and sequences for them to orient them. If you flip them, you can make really funky proteins that would try to like do a loop, and then they were supposed to go into another loop, and we would try to break them, see if we can get them unstuck. It was pretty idea. You had one.
Oh no, I was just saying.
So yeah, I'm gonna play around. I'll give you guys questions about the peptide sequences. Those are pretty cool. All right. So here we're gonna be talking about two different types of mechanisms of getting proteins out, which is gonna be the constitutive, or we're gonna have the regulated pathways, right? In this case, it's soluble. So in this case, it's like there's no mechanism really controlling it. You have your protein, we can dump them out, and it's coming right out. In this case, right, the big thing is gonna be right here, where it's like the dumping of that chemical, dumping of that protein, dumping of those enzymes is being regulated by some external factor, right? So here you can regulate it on multiple levels, which is you can make it when you make the vesicle itself, activating the transport of that vesicle. So remember, we have to depend still on those microtubules to carry us out. And the other one is membrane fusions. So these membranes have like these really fancy mechanisms of the bubble is getting to the edge. And when it gets to the edge, there's this thing called T snares and B snares, which literally look like little wires hanging off this thing. And there's another wire set on this thing. They hook onto each other, and as they squeeze tighter and tighter, they basically like make this smash into that plasma membrane. And that's what makes it dump out its content. All right. So as we're doing that, this kind of has a little bit more color to the drawing we saw before. Basically, those arrows coming in and out. Here, it's just giving you a little bit of a nicer color code of when something's actually coming in versus when something is leaving. Right. So if we start our story as a protein, we start on this side, right? DNA, we make RNA. RNA, we're going to make it into a protein. That protein, we're going to carry it, trap it here, send it into the cis side of the Golgi. We'll get more sugar modifications. Each time we're putting in these vesicles. So in a vesicle, we're trapping the protein we want, send it to the next compartment, right? Do our extra modifications, send it to the next compartment. And once we're ready, we trap it in the last compartment and we can send it out. And then we can go to the plasma membrane to secrete it. It'll basically be on the extracellular side. We can do what's called rate, which is the endosome stuff, right? Which is these guys basically have to mature. So early endosome, we can get it to there. If it's a protein that we make to mess up other factors, then it's going to come from this side, right? The other side is going to be from here. It's like we can also get them from things that we want to digest or eat. We can get them from the outside of the cell. When those things are coming together, we build this mature endosome, and eventually that leads to the lysosome. So let's look at how it's going to break all this stuff down, right? So again, for an endosome to work, we need things coming from two different things. Enzymes from us that eat, and then we need to import whatever proteins or stuff that we're going to eat. All right, so in here, what we're going to find are going to be basically our big eaters, right? So for this one, emphasis on the ACEs. Nucleases, that's going to be stuff that eats RNA and DNA, right? Proteases, we're eating proteins. Glycosidases, right, what we're eating. Sugar, right? Glucose, sugar. Lipases. Right?
Lipids.
Thank you, sir. Yes. Phosphatases.
Phosphate.
Phosphatases, yep. Sulfates, sulfates and phospholipases. Basically, the whole thing is we're gonna have these enzymes and we're gonna start cutting things up. Right? So big catalytic function for burning things. With that, this compartment, we're also playing with it in the fact that we're really bumping up the hydrogen concentration, right? As we're doing that, we're basically decreasing the pH and you're making a very acidic environment. Low pH is basically gonna help us cut things. Like when we think about it with food, low pH also helps us cook. Like I don't know if you guys ever done like where you get shrimp, you put lemon juice on them and then you can cook them like that. Or with onion, or where we like cook food. That acid thing is gonna help us break the bonds and basically digest the food. So in this case, I'm gonna skip that one. I'll come back to it a little bit, but in this case, the big thing is in these lysosomes, we're sending out proteins that are gonna go be doing this function, right? Eating the things. And their function is basically regulated by the pH itself. So that enzyme is not active, aka our protease isn't cutting proteins until we get it into the light compartment. In there, we're changing the pH. Now it's an active cutter, so we can eat here. The thing you don't want would be for you to make a protease, let it be inside your Golgi apparatus, and it's like all the other proteins you've been working hard to make. That thing would actually start to eat them. Right? It would take down your efficiency so much.
What happens if the lysosome malfunctions or if it has like a mutation? It can't function. So like are the enzyme going to break the substance down? Like what's gonna happen?
There's two ways that they can malfunction. One of them is like hyperfunction or hypofunction. Hyperfunction, it works too much, it's gonna mess up good things, and you're gonna be basically like your cells aren't gonna be functional enough. And if it's not active enough, you're gonna build up waste. I think that's related to like Parkinson's and all that stuff. You can't really digest all these bad or misfolded proteins, and your system's not gonna work properly. Like misfolded protein like prions, another bad one. If you can't eat those and digest them, get rid of them, you're pretty much gonna hurt yourself pretty bad. So yeah, good question. So, Alright. So for this one, I'm not gonna go deeply into the sugars, just know that our enzymes, I'll make sure I look at enzymes, are really dependent on that pH to actually, yeah. Alright. Cool. Send me, give me your little cards, and I think we're good for today. Unless we have other questions.